Well, hello everybody, and you are really welcome to this first special webinar from the CHET and the history committees together in order to give a historical perspective as well as a current view of the state of the art in a subject that is important for neuroscience. As current chair of the history committee, it's a pleasure for me to introduce the two speakers, and also a pleasure to collaborate with Patricia Gaspar, that is the chair of the CHET committee of FENS. So the first speaker, as you know, the webinar is entitled Dendritic Spines as Learning Hubs, and the first speaker that we have is Javier DeFelipe, that is a research professor in the Spanish Research Council, Consejo Superior de Investigaciones Científicas, in the Cajal Institute, and his laboratory is in the CTB, that is the Center for Biotechnology, and in a common institute between the Universidad Politécnica de Madrid and the Cajal Institute. He's an expert in the brain cortex, an expert in dendritic spines, an expert in electron microscopy, an expert in brain plasticity, and also an expert in the history of neuroscience, and especially in the work of Cajal, and especially in the work of Cajal in all these subjects. So Javier will give us a general overview of the historical performance to describe the dendritic spines. And then Valentin Nägerl, that is a professor at l'Université de Bordeaux in France, and works in CNRS, Centre National de la Recherche Scientifique, at the campus in Bordeaux, will give us a summary of his work that is really a cutting-edge work in the role of dendritic spines in all these stories related with learning, memory, and stabilization of synapses. So it is a pleasure. And now, Patricia, you have time to talk.
Just to complete, so we're very happy to have so many people attending this. So this new format of history and historical perspective of modern science, I think is exciting. I just wanted to let you know that during the session you can ask questions with the Q&A. So please do so as you see them, and then we will, at the end of the two talks, take your questions and you can ask them to the speakers. Also, another thing that we are interested by you is other themes that you would like to see discussed in this series of webinars organized by the Committee for Higher Education and Training and History Committee. So without further ado, I think we first hear Javier DeFelipe talking about history. Thank you.
Thank you.
So I share the mic. Let me see. Can you see it? No.
Yes.
Oh, okay. Let me see here. Can you see the first slide? No, I cannot see it.
Javier, you should see your slide presentation, yeah?
Can you see it now, right? It's okay?
Excellent.
Okay. Well, first of all, thank you very much for this invitation. And it's an honor and a pleasure to participate in this webinar. So today I'm going to make a introduction about the dendritic spines, the early interpretation of these elements, and one main conclusion is that this is the discovery of the dendritic spines is a very interesting chapter in the history of neuroscience, because it shows that progress in the study of the structure of the nervous system is based not only on the emergence of new techniques, but also on our ability to exploit the methods already available and correctly interpreted the microscopic images, as we will see with the analysis that Cajal and Golgi made with these cells structure. Sorry. So I would like to first start with these important features, that is the two important adversaries for a histologist or anatomist is the smoothness and the colorless of the nerve cells. So the discovery of when you see, for example, this brings a section like this at a high magnification, just only microscopically you can see here the white matter, the gray matter, and it's practically transparent. There are no colors. So the advance in our knowledge of the nervous system depends on the development of light microscopes, because it's the small size of the neurons, and the development of staining techniques to visualize the structure, because they are colorless. So this is at the very beginning, the staining of the nervous system were using very primitive stainings like this carmine, that is you can visualize here. Can you see the arrow here with the mouse? Can you see it?
Yes. Yes, it's fine.
Okay, so you can see here the Purkinje cells stained with this carmine and saffron. So you can see a very little details of the structure of the neurons. In 1852, there was this paper, a book published by Kelliker, who was one of the major histologists of that time, in the times of Cajal. And this was a book published in 1852. That was when Cajal was born, and that is a classic book, because that was the one that was used most students of medicine at that time. And you can see here the different types of neurons in the cortex. You can see a very primitive view of the types of neurons in the cerebral cortex. There are cells like this one that there are no processes, just only without any processes, as I say. And that is very interesting, because there were many papers at that time talking about that there are two major types of cells, one with processes, another without processes, like you can see here. And also, this is the fibers, a fiber that was shown labeled here and drawn by Kelliker. And interestingly, at that time, people didn't know that they are coming from neurons, or they represent like a crystallization of the gray matter. So they don't know. So the structure of the nervous system was very primitive, and very little was known about type of neurons, connectivity, and so on. And this is only in 1852. That was not many years ago, and that is when Cajal was born. So this is the hippocampus before Golgi. This is on the right. You can see this very naive way to draw the hippocampus with those techniques. And here we can see some of the major scientists at that time: Golgi, Viccioreccio, who was the mentor of Golgi, Kelliker, Fusari, and Perroncito. They were in a meeting talking about the structure of the nervous system at that time. So in 1873, that was a giant step for neuroscience, because Golgi introduced a new technique to visualize the nervous system, neurons. So it was a very simple technique, just only put the brain slices and harden with potassium dichromate, and then put silver nitrate. I'm not going to enter in this in details because, but this is a very interesting discovery because it was by chance, the discovery of this technique. So for the first time, it was possible to visualize in an histological preparation all the parts of the neuron, like the soma, like you can see here, the dendrites and the axon, all in the same slice. So you compare the Purkinje cells visualized before the Golgi method with Carmine, and with the Golgi method, you can see really the advance. So the discovery of the reazione nera, as Golgi called this reaction, it represents a major step because it represents the beginning of the study of the structure of the nervous system in detail. So, and importantly, only a small portion of the neurons were labeled with the Golgi method. You can see here on the left, this is a Golgi stained neuron section, that has been counterstained with Nissel, where you can see many these dots, blue dots, are the cells that had not been labeled. And I remember when I started to use the Golgi method, the Cajal Institute, many years ago, I say that it's a pity that with the Golgi method, only a small portion of the neurons are labeled. And then Facundo Valverde, that was there, I was in his laboratory, he said, no, that is it's a fantastic thing, because if you stain in a small portion like this, there's only 20 cells that we can see here, then it's impossible to visualize the different types of neurons, the connectivity and everything, because the complexity of the neurons. So you need to label selectively on very few neurons just to try to distinguish between different types and the connectivity. So the advantage of the Golgi method was only a small portion of neurons were labeled. This allowed the characterization and classification. Here you can see the typical pyramidal neuron. You can see it's called pyramidal because they have pyramidal shape with a typical apical dendrite. Also interneurons like this one, and also the detailed morphological analysis like the discovery of the dendritic spines, and we will talk later, and the possible connections, because you can follow the axon of the cells and just to analyze the possible connections. So with this technique, it was possible to do all of these things. That was really a dream for the histologists just before that method, the Golgi method. So this is the first illustration of the by Golgi of a Golgi impregnation. That is fantastic. That was in the Cajal Institute. I found this picture in a publication from the olfactory bulb of Golgi. I was looking for the very first publication, the very first illustration of Golgi, and that was this one. So this is amazing because just overnight you can see from nothing, then you can start to see here different types of cells, pathways, possible connection, different types of neurons, glial cells, and many other elements. So this is amazing, that you can see this big change. So this is the illustration by Golgi of Golgi impregnated preparation with the hippocampus and the cerebellum, and this is pictorial, and that is in Golgi with the same with the Golgi method, and you can see that Golgi, the structure of the hippocampus, will start to give details analysis of the morphology and the possible connectivity. So an important aspect of this story is that the illustration of the Golgi impregnated preparation were not dendritic spines. You can see all the dendrites of the Purkinje cells, and in the C1 pyramidal neuron, for example, or in the cortex, the cells, there are no spines because they thought there were artifacts. I will show you later. So in 1887 was when Cajal started to work with the Golgi method, and after a visit to the laboratory of Luis Marro. I'm not going to enter into details because I will not have time to explain this, but was by chance that Golgi, Cajal met Cimarro, and then Cimarro told him that there was a fantastic new technique that you can study in very detail the structure of the nervous system. Cajal, when saw that, he was very impressed and said, as soon as I come back to to Valencia, I will start to do to use this technique to study the nervous system. So in 1988 is when Cajal entered into the scene, and this is the first illustration with the Cajal with the Golgi method. And it's important to emphasize that this publication was 15 years later of the Golgi's publication. So for many years, the discovery of Golgi was not applied or was not used for many. The majority of scientists, they don't use it because of reasons I don't have time today to explain, but it's very interesting that for many years that was not exploited. But when Cajal saw this technique with the hands of Luis Marro, he was impressed. And only 1 year later, he published this paper, and then with two major findings. First, Cajal reached the critical conclusion for the neuron theory that axons and dendrites end freely. But this is something that beautiful part of the history of neuroscience also. But also in that paper, he named and described the system of dendritic spines in the same paper. So this is the Golgi preparation made by Cajal. This is at the Cajal Institute. You can see these beautiful basket formations, and these are the dendritic surface of the Purkinje cells. So for the first time, Cajal named and described the system of dendritic spines. So when Cajal saw through the microscope the Golgi preparation, he saw these spines and the axons, and he was looking for the possible contacts with the axons. So he thought that the axons would make connections with the dendritic spines. So here is in 1893, sorry, when this is also original preparation of Cajal with different techniques of the Golgi method. You can see these are the thorny excrescences of the CHET of pyramidal neurons, which is beautiful. These are made with, of course, with a modern microscope. This is a preparation from Cajal. So in this paper, Cajal say that the spines are the points of contact with mossy fibers from the dentate gyrus. So he started to make this, given the importance of the spines. So in the cerebral cortex, he said that the axon of the pyramidal neurons, they establish synapses, so connections with the spines of other pyramidal neurons, and that the dendritic spines of the pyramidal neurons are the major target also of the axons in the cortex. So he proposed that there are key elements in the brain function, So and this is very interesting because these are the spines for other investigators represent artifacts produced by the Golgi method. This is like a precipitate. So you can see here this Golgi preparation drawn by Golgi, but Schifferdecker and Corsey, and this other is from Meyer, where you can see the dendrites or the pyramid neurons are smooth. There are no spines. So here you can see a indication. So the important thing is that the Golgi and Cajal use the same methods, the same techniques, and the same microscopes. So you can see here this Golgi staining by Golgi, and this Golgi staining by Cajal. And in this preparation of Golgi, you can see also the dendritic spines. So the major difference between Golgi and Cajal and other authors is that Cajal saw differently. So it's what I said at the beginning of this talk, that sometimes you have a new technique, but not only is important to have a new tool to analyze the nervous system, but also is the way that you use it and how to interpret what you see with the microscope. So because Cajal was tired of this, because he said that the spines are very important, are real elements, and people don't believe it, Cajal then used other techniques like the methylene blue that was published in 1896, where you can see exactly the same thing that you see with the Golgi method, that the dendrites were covered by spines. So, as in dendritic spines were consistent staying with both the Golgi and the methylene blue method, that is completely different method. In specific regions of the neuron, Cajal interpreted this as a clear evidence of the existence. So this is the Golgi method again, and on the right is the methylene blue. So the spines are true anatomical dispositions. So in 1896, so Cajal said the following: dendritic spines are constantly present at the same regions of the dendritic carboxylation, no matter what animal is studied, and are always lacking at a certain size, such as the axon initial segment, cell body, and origin of the thick dendritic processes, the proximal process of the dendrites. So he said the conclusion was that spine must be key elements of the structure and function of neurons. And you can see here on the right, so this is the employing the Golgi method. Cajal presented a complete morphological description of what is now recognized as a typical pyramidal cell in the cerebral cortex. That is to say, you have the apical dendrite, and the terminal dendritic shaft, the basal dendrites, axon collaterals, and axon entering the matter. And that is important because now is the picture that we have about how it looks like the pyramidal neuron in the cerebral cortex. And the distribution of the spines is critical because, as Cajal said, there are no spines on the cell bodies, axon initial segment, and proximal portion of the dendrites. And that means that the glutamatergic synapse on the pyramidal neurons are avoid these regions. These perisomatic regions are connected by GABAergic connection that we know now. And this is very important from the point of view of connectivity. So, and this is a very interesting thing. This in 1901, that preparing the other day a paper about these things, I realized when I go a higher magnification that Golgi started to drop spines, as you can see here on the right, in 1901, before no. So that was after Cajal insisted that there was real dispositions and used different techniques. So Golgi had no other chance just to accept that they were real, but he was not, he don't say anything in the paper. Just only draw the spines, but he didn't mention about these things. So he forgot it. I said, okay. So you can see here, it's amazing because when you see this, you see all the drawings of Golgi with no spines, and then start to draw the spines, but he didn't mention the spines in the talks, in the paper. So one very important thing is that Cajal, that also introduced the term plasticity in the brain about the structure of the neurons, he said that as opposed to the reticular theory, the theory that cellular processes could develop fine algorithm seems not only the most likely, but almost the most encouraging. So he was thinking that the cells must be dynamic, no? And he said this famous phrase: any man can be, if he wants, the sculptor of his own brain, because the brain was plastic. And he started to use this word also for the very first time, talking about the structure of the nervous system. And just to, I'm going a little bit fast, but it's okay. So the question at that time also is that if the neurons maintain a fixed structure, or they can change their motility. And that was a very interesting question, because people start to think that, for example, you have here these drawings of the spines. Many of the researchers were thinking that it was impossible that a neuron have a fixed structure. These spines are always the same through the whole dendritic arbor. There are thousands of spines in a given neuron. So this is because we are using fixed tissue. This is like to take a picture of something that is moving. So the people start to think, the researchers at that time, that probably this represents something that was dynamic, and they were moving the spines, and they were changing because these different morphologies represent different moments of the life of the individual. So in the Cajal say, it seems very likely that the spines represent points of contact of the or of the resection of currents. The retraction would cause the individualization of dissociation of neurons. On the contrary, the state of activity will be correspond to the joint, to the latch. So you can see this was one of the ideas they have. For example, when you are sleeping or you are doing something and then you start to do something very fast, they say there might be the spine that were involved in these connections and things like that. No? So as a summary, at that time, this is amazing because now Valentin will talk about these new modern views or techniques to visualize the neurons, but in the times of Cajal, this is from a review I published in 2006. There was the hypothesis that they already worked many years ago. It looks like a paper from a recent article. So they have activity. In activity, the spines are moving, they are connected. Also, the glial cells is another hypothesis, was the processes of glial cells intervene between the dendrite spines and the axons just to inactivate, and also that before sustained activity or learning or other things, and after sustained activity or learning, you can see an increase in the number of spines and of the connections. So these are the things that we are at present working with modern techniques, but some people don't, researchers, in particular young people, think that when you are talking about this, you are talking something that is very new, but that was also at the beginning of the history of neuroscience with Cajal and Golgi. That was the idea that they were working. So, and this, just to finish this first part, I'm going to talk because I don't know if I will have time to talk about continue with the study of the electron microscopy and other plasticity and other things. I would like to finish for this first part with this thing that this letter that I call the last lesson of Cajal, with the final lesson, because this is a letter that he was writing in his bed when he was very sick. In fact, that was writing in the 15th of October in 1834, just 1 day before, 2 days before he died. And he was reading a letter from Lorente de No, his pupil, famous pupil, and then Cajal was reading a famous paper about the hippocampus that Lorente sent him, and he made this beautiful drawing of the spines, and he say only 2 observations. I'm going to read only the first one. Spines, not that they are not sharp, irregular excrescences, but genuine spines terminating with a ball. The pedicle neck is sometimes too pale to distinguish. So because Lorente was drawing the spines, but we know the exact way that Cajal thought that should be drawn. So I say this is the last lesson because even when he was, just as I say, before he died, still he was working until the very last moment of his life and still teaching and talking about the spines. That was something that for one of the major discoveries that Cajal had and was talking for many other things. So, and now I think, Valentin, I think you should continue, and then if I have time, I will continue with the other part of the talk, if we have time. Thank you.
Thank you so much. It's so nice, thought-provoking. I know that Cajal wanted us to work on rabbits, yeah?
Oh, yeah.
Than mice.
Yeah. Yeah. No, because he was thinking that the rabbit was more higher mammal. I mean, it was more complex, you know.
Probably
so
right.
Use rabbit and then do this, and then the funny thing, because Golgi, Lorente was drawing the spines like spines of a rose, you know, that came the name from the roses, no? I said, no, you have a ball in the head, you know.
Thank you very much. Now I
have to read.
We'll have a general discussion, and the floor is open to Valentin.
Okay, yeah, I'll share my screen now.
if you could open your presentation.
Yes. Okay, can you see this?
Yes.
Okay, all right, yeah. Yeah, I'm very happy to also participate in this webinar, and very proud, honored to follow up on Javier. Yeah, I really enjoyed to hear about the cerebral gymnastics, so another term for synaptic plasticity, of course. And I wasn't so aware of how people back in the day were were sort of willfully blind to these structures, that there was really this whitewashing going on the dendrites, and even though Golgi was seeing them, but he was ignoring them blissfully, and then slowly they crept up and they became sort of undeniable. So that was really fascinating to hear. Yeah. So yes, I will be happy to review, sort of take up the historical train, and we go up to more recent developments, and then you know, and then have a discussion on, you know, Quo Vadis, the field of dendritic spine research. So we have, I should start also my timer here a little bit. So if you type in what is the functions of dendritic spines, you get some very reasonable answers. Pitch perfect, GPT style. But this is obviously the easy part, and a lot of the things that you see here on the functions of spines are actually curiously were predicted in many ways already by Ramón y Cajal. We are really still in the age of sort of validating Ramón y Cajal, it seems, in many ways, and it's great to be now in the 21st century to see that the sort of this enthusiasm about dendritic spines carries on, right? And we have a lot of techniques, and that is that's the harder part. But we have these techniques now that allow us to really push these concepts much further and really put sort of meat on these hypotheses, right? And really distinguish ideas, speculation, conjecture from really established fact and you know, ground truth. So certainly we all, as neuroscientists, know how important all these different labeling techniques were already back in the day, as we've just heard, right? The Golgi and methyl blue technique and so on. Now it's transgenic labeling using fluorescent proteins. We can not only label specific cell populations, but really with molecular specificity, particular targets inside of cells or even outside of cells, on the membrane of cells. We got a big boost last 20, 30 years out of method development, microscopy development, hardware, technology development, confocal and two-photon microscopy especially. More recently, last 10, 15 years, we've seen the advent of super resolution microscopy, and spine research is currently really taking off because of that. Again, it's sort of going through a renaissance because of the development of super resolution microscopy. I think in the last 10 years, we have really seen biggest progress from the area of protein chemistry, protein engineering to build, to engineer new biosensors, smart biosensors, and also actuators, GCAMP or channel rhodopsin, for example. And then we have seen incredible progress in terms of the ability to do in vivo experiments in intact tissue, sort of in situ, in the compartments that matter, that we care about, so spines, the synapses, yeah, by way of cranial windows that allow us to directly access and watch in real time, if you will, on what's happening at this micro scale. And then field has benefited also from the development of various model systems to study brain diseases. And then last but not least, we're just seeing now the several revolutions in the area of analysis, computational approaches, not only to enhance what we make of the images, but also to produce better images, right? The computer is being sandwiched between the microscope and us to actually now also guide the microscope to have sort of a type of smart microscopy by way of adaptive optics and other closed loop feedback systems to improve the imaging that we do, and then help us process the imaging and analyze the image in a much more efficient, reliable, and high throughput way. Yeah. To boot, all these technological changes are highly compatible. They're synergistic, and they're getting better by the day, by the week, every year. We're seeing really significant progress. Yeah. So all this has allowed us to get new insights into the morphology, but also the diversity that's becoming more and more apparent. They're not all spines, all synapses obviously alike. We get now, because of the imaging, the non-invasive imaging, we get ideas about the dynamics and also the function that way, and the plasticity of dendritic spines. Yeah, so I will be happy to review that. So just more by way of background. So we have the challenge certainly is the sort of space-time challenge, certainly the challenge in spatial resolution, to get enough of it to reveal neurons on the scale of micrometers and neural circuits. But then if we really want to see synapses and look inside of synapses and get some pixel resolution inside a spine, we really need to have nanometer resolution, right? The volume of a spine is around a femtoliter, or could be a fraction of a femtoliter. The volume of a segment of a dendritic spine neck is around an attoliter. And just to give you a feel, sort of the relationship between a femtoliter and a liter is like the relationship between a liter and the volume of the Mount Everest. So then the relationship between an attoliter and a liter would be like a liter and going to the whole Himalaya or the whole Alps. Yeah. So this is where we obviously we don't have any intuition anymore. This is way beyond what we can imagine. Yeah. Let's say at the beginning, but now with microscopy we can peek into it. We can see the circuits. We can see the cells. We can peek into synapses, and we have imagining things. But now we're getting experimental feedback on actually what's happening on this scale, and we can see the morphology nicely, begin to see it really nicely, as well as the molecular composition in its dynamics. But still, as these techniques are relatively new, there's many questions on how these structures are really built and maintained, and how they may also break down in disease. And then also the relationships between these different levels of analysis are at best very cursory, right? To go from the circuit level down to the molecular level is still hugely challenging. Likewise, to go from the circuit level up to the macro scale and the cognitive level or behavioral level, that's equally daunting. Now, in such a difficult faced with such difficulties, it's good to get wise advice, and I very much like these words of wisdom here from Sidney Brenner, probably familiar to a lot of people, on how we can hope to progress here in neuroscience faced with such a challenge, and that is to focus on new techniques that allow us to make new discoveries, and then come up with convincing ideas to put it all into a cogent, coherent theory. Now, mapping out and understanding the function is usually very hard, because it depends on so many factors, especially in the brain, where the tissue is so fragile, so fleeting, yeah? Where you have to really zoom in here into this nano domain. This is certainly the micro domain. So if you can't do the function, you can at least start to do the structure, and that is, I think, in many ways now you're seeing a renaissance of neuroanatomy now by way of these connectomic studies that focus on that. Yeah, getting a snapshot, albeit a nanoscale snapshot across the entire brain, reconstructing these synaptic circuits. And then finally, I think it also helps to have a good theory, to actually, to have some some hypothesis and not just do data mining or collecting data ad nauseam. But I think it's good to have a little idea on what could be these structures good for. To have a functional approach, to have a physiological sort of reasoning or some sort of evolutionary reasoning, and not be so completely unbiased, because then I think we'll be lost in sort of an infinity of possibilities, and we really have to zero in. Now, physics typically has very good theories. They can look ahead some decades or 50 years or so. Neuroscience doesn't really have a whole lot of good theories, and it's more sort of in hindsight that we cobble things together. But I think still it's worth to develop a strong hypothesis. Now, I just want to quickly go through some little bit of history as well. Not reaching as far back as Javier, but this whole field of spine plasticity already started some 40 years ago in the 70s. Really trailblazing anatomical work from an electron microscopy work from Harreveld, lab of Harreveld and Eva Fivkova, postdoc at the time, talking about swelling of dendritic spines, morphological plasticity, and how this may underpin synaptic plasticity, and also here already talking about spine necks that may be altered during activity-dependent stimulation, and also speculating already or going into molecular mechanisms, looking at protein synthesis dependence of these changes, and talking about how changes in calcium dynamics, calcium liberated from the spine apparatus may subserve these things. Now this is an early literature that I think most of us, also the audience, probably not familiar with, and it has kind of gotten buried because also at the time it was very sort of haphazard and very anecdotal. It was electron microscopy, which doesn't lend itself to do really dynamic studies. But yet, these were already, the concepts were certainly around and alive. But really there was a big, I would say, major second life or revolution in the field happened in the 90s with the advent of two-photon microscopy, which provided the first evidence sort of in real time of spine plasticity in living tissue. And these are two breakthrough studies then in the end of the 90s using time-lapse two-photon imaging to delineate at the level of the single dendrites the growth of either spines or filipodial-like processes, right? And these studies were done in sort of longitudinal, over minutes and hours, and paired with functional recordings and stimulations provoking the acute generation of these processes. Then a few years later, also still with two-photon microscopy, but now with maybe more improved optics, we could also contribute to our understanding that this plasticity is a very consistent phenomenon. That spines can grow rapidly on the timescale of minutes, and can do so in a bidirectional manner. They can not only grow, which has been described before, but also they can retract, shrink in size, and that this is nicely correlated with various induction protocols of synaptic depression or long-term depression, LTD. Right? And then another landmark technical development came from the lab of Haruo Kasai in Tokyo, University of Tokyo, who developed the possibility to target individual spines in a very specific and arbitrary way using two-photon glutamate uncaging, which allowed them to observe in single spines that they increase significantly in volume as they undergo functional plasticity, right? And this really heralded a new era in the structure function analysis at micron scale of dendritic spines. Then in the following years, there was already then there were these breakthroughs in the labeling and the in vivo microscopy, which produced a lot of studies looking at now in an intact animal, looking at experience-dependent structural remodeling. And there's already here a review cited already in 2008. The field had blossomed to the point that there were already dozens of papers on that subject, right? And then also I want to highlight here, point out a study also from the Kasai lab that already showed or hinted at evidence, let's say, that really this spine plasticity is not just a curiosity, something that we observe in vitro, or maybe it's just an epiphenomenon, but maybe really functionally important. And they showed this by erasing so-called memory spines that were generated in a learning paradigm, and could see that this actually is important for the maintenance of the learned behavior or the learned motor skills in that case. Then a few years later, we were part of this development of making use of super resolution microscopy to just study the dendritic spines. And first and foremost, the technique that is particularly useful here is STED microscopy, which you know is a laser scanning technique that functions much like confocal two-photon microscopy, but has the benefit of using a fluorescent spot that can be greatly reduced in size. Yeah, instead of having a point spread function of around 250 nanometers, one can reduce it effectively to well below 100 nanometers, even 50 nanometers or smaller, right? And with that, it becomes possible to really see not just the presence of spines and see them in a rather fuzzy manner, but really get down to, I would say, the anatomical ground truth, not necessarily the molecular ground truth yet, but the really delineate the fine nanoscale morphology of spines. Yeah? And in this video here, you can see that we can really get the 3D view now of spine morphology in 3D tissue. At the time, just in the early days in brain slices, but increasingly also now in vivo. And this is an example shown here where we could use two-photon and then two-photon STED microscopy to get a much sharper view on spines now in the hippocampus, which you know is deeply located, deeply situated area in the brain that's hugely important for memory processes. And here you can see the benefit a number of years ago already we could achieve by switching from a regular two-photon system over to two-photon STED microscopy, which gives much better readout of the actual number of dendritic spines. And that allowed us to quantify over a number of days here the dynamics of spines on identified stretches of dendrite. And you can see here that actually over just a few days, the dendritic morphology becomes hard to recognize because of the high level of structural turnover, where some 40% of the spines turn over within just 4 days in this area that is known to be very plastic functionally and important for plasticity memory. And then now sort of the state of the art now is now with 3D STED microscopy in vivo, where we can obtain images like this. I'll show you video also that is that really now where we can in vivo basically create reach an image quality, a spatial resolution that compares to what we used to get only in brain slices. So then a number of years ago, we could use STED microscopy and combine it with two-photon glutamate uncaging to target really individual synapses, induce plasticity, and have as a functional readout also electrophysiological recordings in parallel, right? And then we could really go into the nuts and bolts, into the details here of spine morphology, and correlate the sizes of the spine head, and especially the spine neck, with biophysical properties. For instance, the diffusional isolation of the spine head from the rest of the dendrite, which very much depends on the neck geometry, as one might imagine. Could also correlate it with an important functional parameter, and this is the spine neck electrical resistance, which we could show varies over orders of magnitude. So not all spines have some, some canonical neck resistance, but it can vary from over 3 orders of magnitude, or up to 3 orders of magnitude, right? And then we could demonstrate using the uncaging technique to target individual spines, that during a LTP paradigm, that the spine neck becomes wider and shorter, actually. Yeah, something that has been hinted at by electron microscopy, the work of Fivkova that I pointed out at the beginning of the presentation. But now we really have direct sort of real-time in vivo or live cell evidence. This has some interesting functional ramifications in that when you consider sort of a regular spine, sort of a normal spine with that features a relatively high neck resistance on the order of hundreds of mega ohms, you can imagine that strong synaptic activation then can isolate the electrical signals very well, which has the consequence that the electrical potential, the voltage signal in the spine head may actually saturate by virtue of the fact that the local input impedance provided by the spine neck is so high that the voltage in the spine head becomes very large and reaches the reversal potential of the synapse. Whereas after the induction of LTP and those structural changes that we have delineated, that we have discovered, when the spine neck becomes wider and shorter, affording a reduced neck resistance, that then the electrical potential will be lower, which is a bit counterintuitive. It will be reduced after LTP, but that reduction has functionally the effect that more charges, more current can actually enter the synapse, because the synapse is not being driven into saturation, and it remains further away from the reversal potential, where it would not conduct any more charges, right? So so then the functional consequence would be then with a reduced electrical resistance, or a reduced input impedance by way of these purely structural alterations. The consequence would be to get a lower voltage in the spine head, but effectively a higher voltage in the soma, because more charges can be transferred before the synapse would be effectively saturated. Okay, then I need to brush up on a few others.
If we want to leave 5 minutes for questions, we're
Okay, then I maybe leave. Then I just skip over this briefly. Just want to highlight, just mention a few terms, and that is sort of really new areas or new concepts that have emerged beyond obviously Ramón y Cajal, and this is afforded by super resolution microscopy, specifically by single molecule imaging. And these new techniques over the last 10-20 years have given us this idea of whereas the synapse or the spine is something rather can be something that's rather structurally stable, although I've shown you now lots of examples where actually this is not the case, where it also is dynamic. But what we know based on these new approaches, that the content of the spines is highly dynamic, right? The synaptic receptors are being turned over rapidly, and they can move rapidly. They can move laterally, and they can move in and out of the synapse. So that's something we can maybe leave for our discussion a little bit. So that's one important new concept that has emerged last 20 years. And another concept, even more recent, last 5 years now, last 10 years or so, is the idea of a transsynaptic nanocolumn, where we don't just consider the postsynaptic side, of course, where most of the action is, but there's also a presynaptic side, of course. And this new concept based also on single molecule imaging and other super resolution approaches is saying that actually a synapse is highly coordinated across the synaptic cleft also, right? Molecules, the presynaptic release machinery and the postsynaptic density are highly co-aligned, and there may also be potential for important mechanisms to elicit and support synaptic plasticity. And then finally, another concept which has also gained traction again in the last years, but over the last 10, 20 years also, is the idea of the tripartite synapse, where it's not just the pre and the postsynaptic compartment that play an important role, but also the glial coverage, especially astroglial coverage from astrocytes that can regulate, can sense activity at the level of single synapses, single spines, but also can influence it. Here's just some images based on STED microscopy of some glial processes that enwrap spines, and they form like a ring-like structures as if to sort of cradle the spine and maybe even exert mechanical traction on it. Okay, then there are new approaches to study spines. I have to go quick now, maybe leave that for the discussion part. Let's discuss this. So before we do so, let me acknowledge most people in my team who have contributed to our work over the last few years. And I look forward to some questions now. Sorry for racing this now at the end, but 20 minutes is shorter than I thought.
Well, yeah, I think, well, I guess there was so much to say for both of you. I'm sorry. I mean, probably these, the format,
it's just a teaser, yeah.
The format is really short, and we had to, it was frustrating for you to compound this. So we have very little time. I have to unfortunately leave you in 5 minutes. I can just mention some of the questions that were raised. I mean, both of you, I think it was very fascinating. I think you have questions that are related to disease and you know, applied scientific knowledge. So I guess one is, you know, what's happening in disease. So maybe this is maybe too broad question to be discussed here. Yeah, so one is, you know, the view of perspective. What is the direction and study of spines going in the future, both in technology and development and applied science?
Mm-hmm. Yes.
Yeah.
So we, as I've listed at the beginning, all these methodological advances, I think we're going to be seeing you know, in two directions. We're going to see progress, methodological progress, and then also some new conceptual insights. One is really link the spine, which is this relatively stable structure, to its molecular composition. And then, you know, we're seeing that there's a wildlife, crazy life inside, you know, zoo of particles inside the synapse, giving life, you know, animating what a synapse does. And we're going to have many more techniques now to do that with more resolution, less invasively, and so on. And then the other direction is to integrate, you know, individual spines, right? We have a ton of them in the brain. We have 100 trillion spines or synapses, at least, in the human brain. And to integrate and bridge the gap between the structure function of individual spines and what they do in as a group, yeah, as 1,000 of them, 100,000 of them, and, you know, 1 million of them, and how they may be regulated by other factors, outside factors, by the extracellular matrix, by glial cells, and so on. And I think that in both directions we're going to get better. And we still live in the age of, in terms of microscopy, it's the age of fluorescence microscopy. I think this still has some strength and some mileage in it, but there will be something afterwards for sure. Yeah, maybe there'll be like an MRI with cellular resolution, with microscopic resolution, yeah, which would be fantastic.
Yeah. One thing also will be important to emphasize the need of make models and simulation because of this complexity of the dynamic structure of the spines. The only way to try to understand what is the significance of that will be to make models. Because if a pyramidal neuron, you have over 20,000 spines, and all of them are moving, changing the size and many things. And just to try to understand 3-4 spines together is really hard. Imagine what happened in the whole dendritic arbor of only one pyramidal neuron, how these changes in different parts of the dendritic arbor, what is the impact in the function of the neuron. This is something that we need models to try to understand better this. And also, I didn't have time to show you the new techniques for electron microscopy, volume electron microscopy, where you can reconstruct hundreds of thousands of spines. So you have the higher resolution, or just to compare the postsynaptic density, the size, and many things, together with the light microscopy. Together will be a major step in the near future to get more data about the spines. But I believe that it will be very hard to transform the data in knowledge if we don't have models and simulations to understand what we are seeing. We have beautiful data, but very nice, but we don't understand very well.
Actually, yeah. So that was a comment I wanted to make, because I think there are many people working in this field of modeling the power of how spines is helping to integrate learning and brings a new dimension to computing, neural computing. So I think
and we know that in Alzheimer's disease and many other diseases, I didn't have time to show you, even the very early studies many years ago, you can see dramatic changes in the morphology of a spine. There are some spines that are with the neck very, very long. The neck was the head very small. And things that at that time people don't understand what is the possible functional significance. But now with the study that Valentin is showing, that it can give you an idea of the functional importance of these changes, something that happened in disease of the brain, like Alzheimer's, as I say, another kind of. So this is a very interesting thing. So the spine is a long story, and we have for many years.
So one question related to this relation between morphology is when Robert Lindros asked if there is if the spine neck modification is enough for learning or activation of extrasynaptic receptors. So I
mean, spine neck plasticity is just one incarnation of synaptic plasticity. I think there's, we know there are a lot of different mechanisms at play, and I think no one can explain the whole plasticity phenomena. But I think the neck has been is something that has been brought up over the decades a lot of times, but it hasn't really because it's so difficult to access, and it's even smaller than the spine, right? So it hasn't really found a lot of adherence, let's say, and a lot of studies. Yeah, but I think obviously the neck being so narrow, it sits at it's a crucial, potentially it can have a major influence on what the synapse does and how it communicates with the dendrite, of course. Yeah.
And maybe also a related question is also for Valentin. I mean, for both of you, is the molecules which sustain the formation of dendritic spines. So I think that's a very large literature, I guess. Maybe too large to discuss here, but maybe you want to give a quick answer.
I mean, there's hundreds of different proteins that are now well known. I think the parts list is kind of known. I don't think we're missing anything. We know what ions they are, what the major ones are, and we know what the proteins are. They are all on the table. It's just how they interact, right? And when they and in what states they are, right? A protein can have some slightly different configuration. Then it has a different function, right? So in understanding how this molecular machinery really works in real time, I think there we are still very far away from getting a reliable answer.
Yeah, thank you. There's a question here. I mean, there's some maybe more theoretical questions, but which is about, what do you think about the theory that the connections are still there, and that rather than reorganization in response to a stimulus, the cortex already possesses connections that are silent until it becomes necessary to activate them, and doesn't need any reorganization. So what do you think about this new theory?
Well, yes, my point of view is that, for example, when you do electron microscopy of many spines, you see that about 4% or something like that are lacking of synapse. So, and the question is that the spines that do not establish synapse at that time, maybe later is going to establish a new one. So that is a possibility. So, but now I believe that these questions can be answered better with doing in vivo studies like Valentin is trying to do, no, with these things. You can visualize in vivo, you can manipulate, and then you can see it. The issue is that at the electron microscope level, when you see a button and the spine, maybe you have very close together, but you don't establish a synapse, establish later or whatever. So, but the real thing is that when you study the spines in any part of in the hippocampus or the cerebral cortex, 4% or something like that, that is a lot, millions and millions of spines, they don't establish synapse. At the moment that you analyze the brain, at that moment, maybe later change, but only you have about 4%.
But in addition, there's also the silent synapses, right? That just have NMDA receptor and don't have AMPA receptors, so they could also contribute. But to answer the comment, the question, also, I mean, we know there is this plays a role. There's sort of the unsilencing of synapses, and then the generation of new synapses that are already the functionalization of already pre-existing synapses, but we know there's also real, genuine de novo growth, right? So then all these things are happening in parallel and not at the exclusion of each other, for sure.
Yeah.
Okay, I'm afraid I have to leave you, and I just wanted to thank you. And I think there are still other questions, with questions about organize. There are many interesting questions here. Maybe you can have a look, and I think the people, I mean, please send your question directly to Javier and Valentin. I'm sure they'll have a pleasure to answer your questions. And I just want to close this by thanking everyone, and you too I'm sorry I had to interrupt your beautiful talks, but yeah, so I think this shows that there's a huge interest in this, and there's a lot to be done. Maybe we'll have to do another webinar on, structural plasticity and so on.
Okay.
Thank you so much.
Okay.
Thank you to you.
Yeah.
Bye-bye.
Bye-bye.
Bye.
Bye-bye.